Because this method determines PD-1-blocking antibody-bound cells using a real blood from patients, the specific subset responsible for anti-tumor effects and immune-related adverse events can be identified. This method needs only a tiny amount of blood sample, and the analyzing process is very simple and quick. It just needs a flow cytometry machine.
To begin, collect whole blood samples into blood collection tubes containing EDTA. Treat each 5 ml round-bottom polystyrene flow cytometry tube with 1 ml of 2% fetal bovine serum in PBS. Vortex for 10 seconds. Then transfer 20 microliters of whole blood samples to the treated 5 ml round-bottom tubes. Add 20 microliters of 2% FBS in PBS and 10 microliters of human-specific Fc receptor blocking reagent. Mix well and incubate for 15 minutes at room temperature. Next, add 500 microliters of red blood cell lysis buffer. Mix well and incubate for 10 minutes at room temperature. Add 4 milliliters of 2% FBS in PBS and spin down cells at 400 times G for 5 minutes at 4 degrees Celsius. Remove the supernatant by aspiration. Repeat the wash and aspiration process. Resuspend the cells in 100 microliters of 2% FBS in PBS and divide into 2 tubes of 50 microliters each.